No. Yeah, no, the grade is pretty much an easy breakdown. There's 4 tests-ish, a final, which is the same weight as another test, and the quizzes. And then you get 5 points for being here. That's pretty much it. So there's no papers, there's no midterm, there's no final, cumulative final. So I'd rather cover more material, in my opinion.
Have, like, a whole semester final.
Nope.
Fantastic.
That's a lot of studying right there.
No, but I'd rather cover more stuff,
Right.
in my opinion. Plus, we here at the community college don't have an official finals week. So to give you a true final, I can't give it to you in an hour and a half, right? So to me, that's that's not a final. That's not a cumulative final in my book.
Our grades close the 21st, right? Something like that. That's what I was talking to the registrar.
I think that's the day.
When's the next semester start?
14th of January, I think it is.
That's a long distance. That's awesome.
I thought it was like the 20th or something.
Oh, he wants it to be even longer.
That would be cool.
Okay, so we kind of left off talking about this, and I think you were all kind of, whoa, yeah, okay. So let's just review antigens and antibodies on our blood cells, okay? Yeah, so remember when we talked about plasma membranes and we talked about the phospholipid bilayer and we talked about all those little proteins on the surface. Well, some of those proteins on the surface of the plasma membrane have little carbohydrate groups attached to them. Oh, that's cute, isn't it? These are antigens. Now, your cells have your antigens on them, determined by your genetic information. Yes? And some of the antigens in question here are coded for by your genetic information. And remember, when we talk about genes and alleles, how many alleles code for blood type? Yep. 3. There's 3 different things I can have on the place on my chromosomes. I'm going to pretend those are chromosomes. There's 3 different things I can be dealt genetically. Yes? I get 2 out of the 3. Only. Always. So these are the alleles you can have for blood type. Or a little one. Eep. Yes?
Oh.
It's a little I. It's just the location. They call it I. They typically name these different alleles with something. You'll see them named a whole bunch of different things in your book. They're giving them basic names like for the red color in plants, R. If it's a dominant and recessive, then we have a big R and a little r. Or we have a Y, or we have a W. They give it a letter as we're talking about it. If you're an immunologist or geneticist, it's going to have a big name. But for our purposes, we're just going to give it a letter so that it's easier to get, easier to understand. But for blood type, and this is an example of what we call multiple allele and codominance, we have more than 2 different choices, multiple allele. These are 3 choices we could get for blood type in our genetics. Yes? The codominance thing has to do with the fact that whatever we're dealt, we're going to produce. Okay? Or not produce in this case. Yes? So there's two different antigens we can see on the surface of our cells. A or B, exactly. Or neither one, neither A nor B. So when they talk about human blood group, we're talking about 3 different alleles you could get dealt. Again, how many do you have?
2.
You only have 2. And those antigens on the surface of your cells are kind of like your immune system's way of knowing what cells are yours. So your antigens on the surface of your cells are yours. If I take your cells and I put them in your body, what's going to happen?
It'll be attacked.
We'll attack them based on the fact that your antigens are different than his antigens.
Or if you don't have any.
Or what if you have the same antigens?
Well, we have the same, okay.
Yeah.
But like the eye doesn't have any antigens.
Well, it doesn't have A or B. There's still antigens on the surface of those cells. But with respect to blood type, it's not A and it's not B. So normally when this antigen, your antigens are here, we leave them alone. We know they're ours, they're fine. We're happy with them. But when the foreign antigens come in, our body recognizes them as foreign and will attack them and make a chemical that will neutralize them, called an antibody. And this is kind of where we left off with our discussion on Monday. So if a foreign thing, foreign protein on some foreign cell comes into my body, I'm going to make these chemicals called antibodies that are going to neutralize that foreign antigen and get rid of the invader from my body. Now we can find these antigens on the surface of bacteria. We can find them on the surface of viruses. Anybody have a cold? Yeah, yeah, thanks for breathing on me. What's happening in our bodies? Yeah, exactly. So a foreign thing, probably a virus, because most of our colds and flus are all caused by viruses. Foreign antigens come into our body, our cells recognize them as foreign, and they attack. They make antibodies to help neutralize them. And they'll make enough antibodies to neutralize them when you start doing what? When you start feeling better, okay?
So why doesn't it just instantly like attack the virus as soon as it gets into the body?
Actually, there are some things that do, but there's not enough of them.
But like, if like we were given blood from the wrong, you know.
So here's the catch. Here comes the catch. This big story was so that you understand why that happens. Normally, we don't make antibodies against antigens unless what?
They're there.
Yeah, unless they come into our bodies and we do this whole response. It takes about 7 to 10 days to make up enough antibodies because we actually have to make more cells that are specific to making those antibodies. That's normally how it happens. But one of the things they don't explain and why it's a little confusing with the whole blood type thing is, for some reason, and we have no idea why, we still don't know, your body is going to make antibodies against the antigen it doesn't have. They're called naturally occurring antibodies. And normally it only happens in this case. We have no idea why. So if I'm type A blood, that means what?
You have an antibody for type B.
Correct. I have A antigens on the surface of my cells. What would my genotype be? And here's where the codominant part comes in. I have two eyes, yes?
Yeah.
And I can pick from those three. I can either be this, or I can be this. Can you see that? Yes? Because no matter what, I'm going to make those antigens if I'm dealt even one allele. There's another blood type, B. So what am I if I'm B? Well, I have B antigens on the surface of my cells. And this is my genotype. Or, you said A and I wrote A. Yes?
Where does the O come in?
If I'm type O, what's that mean?
No.
I don't have A and I don't have B. Well, I don't want you to say no antigens, because not to confuse the hell out of you, but you still have antigens. They're just not A and they're not B. You can have a whole bunch of other type of antigens, and your cells do. So I don't want you to think there's no antigens, but with respect to blood type, there's no A's and there's no B's. So what's your genotype going to be? Exactly. And then the other one is this guy. And what's their genotype?
B.
Yep, that's it. Because you only get two. If you got both, one's A and one's B. So I have genes.
So what are the antibodies?
So remember, I'm going to make antibodies to the antigens I don't have with respect to A and B. So in this person's plasma, I have anti-what? Correct. In this person's plasma,
Anti-A,
yep. How about this guy?
none.
Both.
Both.
Because I don't have A or B.
That's why O is universal. Can't take.
So what's that?
That one doesn't have any.
That's the guy that doesn't have any because he's have both A and B.
That's the universal taker.
Now, catch. When you get blood, if I give type O, you said it was universal donor, right?
That's the universal giver.
Yeah, donor. Give to. Yep. If I give whole blood to type B, what's going to happen? They're going to croak. Yep. That's what this little chart talks about. So because I have in my plasma anti-A and anti-B antibodies, if I put that plasma into my patient B, this is going to be a serious problem. But you know what you get when you get blood? You only get the cells. Then when you donate blood, they take your whole blood, which is plasma and cells, and they separate it. They do that at another place.
How do you do that? Do they spin out the plasma?
Yep, they spin the blood down, they take all the cells out, then they actually wash the cells several times to make sure there's no plasma left. And then they put it in saline, normal saline. The cells are only going to last for X number of days. Can't remember what that number is off the top of my head anymore. Huh? Yep, they can use the plasma for other things because there's a whole bunch of different chemicals in plasma that they can extract and use for other patients. But when you get blood at the hospital, that's what you're getting, cells. Okay? So O cells, and not all O cells, only O negative cells. Because there's another antigen we have to worry about, D. So if I'm A positive, I also have these guys on there. If I'm B negative, I don't have those guys. If I'm O negative, I don't have the D. If I'm AB positive, I also have these guys.
So, an A negative could take blood from an A negative person, right?
An A negative could take blood from an A negative, yeah.
Positive could take it from A negative.
Yes, they could, because there's no antigens that are going to disrupt that person. But an A negative person wouldn't take it from an A positive person, because what's the D going to do to them? It's going to cause them to make antibodies against the D antigen.
That sucks. I won't be able to receive blood from anybody besides A negative or O negative.
Same here, because I'm A negative.
Yeah, me
Yep.
too.
Me too.
You have
So,
two. I only have one. O negative. That's it.
That sucks.
yep. That's worse. That he can give his blood to everybody, but
That's why I got you.
so that's what this little chart shows us. And when we test to see what blood type you are, we actually use the antibodies. That's what's in those little tubes.
We did that
Yeah.
today.
Okay. So did you do that in lab?
Yeah.
Perfect.
Not yet.
Did it make sense now?
It didn't work quite right.
No.
It didn't.
No vials.
She didn't mess up, but like the chemicals were kind of like old. I guess.
Old.
And
Okay. She should have ordered new chemicals.
Well, she found
I'm glad
out
you said that.
once, and
Keep it in
then,
mind.
Because I know I am A negative, but it was coming up that I was O negative.
So what did you see for clumping?
Nothing.
Nothing.
So when we redid it, I was A negative.
So it was the chemicals. Yeah. And that's what's in those little vials. That's the antibodies. If I put the antibodies and the antigens together, they're going to clump. So that's what this is all about. So when I don't see any clumping versus clumping, and this is the different genotypes we would see. Okay? Everybody good with that? Yeah? See, it's nice to do this stuff in lab, right? Okay. Pleiotropy is when one gene has control over other genes. And we see this with different characteristics. Sickle cell disease. That is where we exhibit that pleiotropy. So, what happens with sickle cell disease is a person has been dealt genetics that allow them or don't allow them to make the proper molecule to carry oxygen in their red blood cells. That molecule is called the hemoglobin molecule. And remember how we made proteins based on what in the genes? Yeah, that DNA code, every 3 is going to give us 1 amino acid. Remember that whole discussion? Well, in sickle cell disease, our genetics make our hemoglobin protein the wrong shape. It causes them, the cells themselves, to be the wrong shape as well. And with these patients, what happens is those misshapen cells aren't going to be carrying oxygen and carbon dioxide efficiently. See, this is what the cells should look like.
So wait, are all their cells like that?
Not all, but it depends. Depends on if they come under stress or trauma, we'd see more of them versus when they're sort of in a remission type of state, we'd see less of them.
So that person can't exercise all the time.
And that's where the trauma comes in. So when they start using too much oxygen up at a fast rate because they're strongly exercising or hiking or working really hard, they're going to tire easily because they don't have the ability to carry as much oxygen. Or when they start producing those cells at a fast rate, we're going to see even more of the sickle cells that are going to allow them not to be able to carry on strenuous activity. So we see symptoms like weakness and pain and organ damage and paralysis in severe instances of sickle cell.
Is that because they're lacking oxygen?
Yeah. They're not getting oxygen delivery to the tissues as efficiently, and those cells tend to die off much faster, so your body has to deal with those dead cells, and that's where we get the pain and the pressure and maybe organ damage. Organs like the liver and the spleen.
Replace that.
It would be in every single cell on your... Oh,
No,
just
just the DNA.
the DNA, not a problem. Just go in there and with our little toolbox and
Put it up on Jack's hands.
we could.
Well, they say they can give babies whatever color eyes you want, so why can't they take sickle cell and use it off the DNA chain?
Yeah, or give you the gene, a normal gene.
Here comes ethics. Is it ethical to do that?
And that's what some genetic engineering is looking at. Viruses, remember I told you how viruses work? Did I tell you how viruses work, or did I tell somebody else? Well, viruses work this way, and we're going to talk about them in Chapter 10. But they inject their genetic material into your cells, and they take over what your cell does for a living. Causes your cell to make more of them. So what if we could put a normal hemoglobin gene into a virus and have it go and inject?
Right,
Yeah, good idea, huh?
right.
Yeah, that's being done.
See,
But we can't do it for everything quite yet, but that's one of the things genetic engineers actually research. How we could maybe give you the genes that you're lacking. Again, that's an ethical thing. So polygenic inheritance, okay? We're talking about one pair, but what if I'm talking about a trait that maybe has not just one pair, but 3 pairs of alleles that are going to code for a specific trait? That's what polygenic inheritance is. So 2 or more genes on a single phenotype. What's a phenotype?
the trait that shows up.
Yeah, what you see. Yes? So this is the most common one used because it usually blows your mind almost immediately when you see the Punnett square. This one is skin color. Your skin color is determined by how many different alleles?
3.
3. Okay? This one is height. I thought it was skin color. Why is it saying height? I guess this is height. So height's the same thing. Skin color is the same thing, by the way. So 3 different alleles, in this case we're calling them A, B, and C, are going to code for height. So how many different alleles do you have total that are going to determine what height you are? 6. Yes? Now remember, we have only, from each parent, we're only going to get what? 1 of each allele. And in the case of these 3 alleles, from one parent, we're only going to get 3. But remember, because of the law of independent assortment, we can get any combination of 3. Yes? So if we have a very short person, or you could use the same exact thing for skin color. If we have a very light person, or if we have a very tall person, or a very dark person. We see that if we cross true breeders, just like Mendel did with his purple plants and his white plants, we're going to cross somebody that has the exact same 3 alleles in pairs. They're homozygous for each of the 3 alleles. Yes? And then we get the F1 generation. Now here's where the trouble comes in. Remember, I get 6, but I get any combination of those 6. 1A, 1B, 1C. What do we get for these guys? The F2 generation. There's 8 different possibilities. Where do they come from? Let's map them out. So we have A, B, B, C, little c. Yes? So remember how I did it before? I get 1A, 1B, 1C. I get 1A, 1B, 1C. You see where I'm going with this? I get 1A, 1B, 1C. You can keep going. Right. But I'm going to get 8 different combinations. I wish they'd write them out for you so you can see that. But 8 different chances, 8 different combinations of A, B, and C.
Are we going to do like huge I-squares like this?
I know, I wouldn't be cruel and inhuman. I might give you 2, but not 3.
Oh,
2 is a little bit easier to manage. 3 is a pain in the keister, right? So, look at all the different combinations. So for height, how do you know what you're going to be? It's a crapshoot, right? My husband's 6'3". I'm 5'5". I used to be 5'5 1/4". I lost my quarter. Huh? I look shorter or taller? I'm 5'5".
Put yourself on the inversion table every night.
I know, that's what I got to do. I got to. So, my kids, what do you think?
average height.
Son's 6'2", daughter's 5'11".
My son is 6'3".
I'm like the little shorty pants in the house.
5'4".
So again, same thing with skin color. So huge variation. The nice thing is they made these boxes lighter and darker. So pretend that same gene combination was a very light person and a very dark person. Look at all the combinations of color you can have. And we're talking F2 generation. So unless that true breeder is there, you don't know what the heck skin color you're going to come out with. Same thing in my family. I'm Italian, built-in sunscreen. My son, same thing. My husband, Irish guy, burns. Well, he doesn't burn. He's actually not too bad. Daughter, hates us all because she burns. So you don't know.
Is it true that like with each generation the average height gets taller?
We tend to, in the human species, we tend to see taller individuals on the average. I mean, if you look at homes that were built in the 1800s, you know, the little dinky, the ceilings were short, the door, everything was lower. We are tending, mutation.
Because attractive people are attracted to bigger, taller, stronger.
I have a son that just turned 14 and he's 6'2".
Woo!
What do they call that?
Food bills.
Molecular evolution. That's what it is.
6'5".
Or natural selection.
He said, you guys want to grow when you're 18 or whatever?
See, my doctor said that, but I stopped growing when I was like 16.
Yeah, and my husband stopped when he was older. So he had a growth spurt in between, like, yeah. he might
Maybe,
not.
yeah, because I stopped growing around that.
Women tend to sprout up faster and stop earlier. Like I was a gargantuan 5th grader. All my little 5th grade friends were shorties. But that was it. I was done in 5th grade, and everybody kind of just, went past me. So,
Well, he went from like really short to over the summer just going.
yep.
Like,
And males tend to do that. Males tend to do that.
I thought they were like older. I thought they were like
10, and he's still a little tiny.
seniors.
Yeah, but you're a little tiny person too. He might have gotten your genes influence. So, what we see in us is not only genetics. That's what we have to remember. What we see in us is a combination of our genetics, what we inherited from our parents, and our environments.
Yeah, smoking, coffee.
Harsh environments, harsh chemicals, sun, all of these things are going to influence what your genes do, how they, what we say in the genetics world, express themselves.
What about twins? Why is one twin taller than one? Yeah.
It depends on the twins. Now, if I have these two girls are what kind of twins? They're identical twins. What's that mean?
They have the same genetics.
They have the same genetics. So when fertilization took place, egg and sperm came together, they made one. And then when it started to divide, something weird happened, and they split far away from each other and kept going. So we're talking the same genetics, two different individuals. Do those girls look exactly the same? There's a lot that are similar.
They don't even look the same age.
But what happened? This one likes the tanning bed. Yes?
Yeah.
Meats less than the other one.
Hmm?
The one on the left eats a little more.
Yeah, she might like the Twinkies a little more than her sister, right?
Yeah, she looks better.
Her hair. Well, the hair is kind of similar. But you start to see different things, moles in different places. How come she doesn't have one there and one there? No. I think she just likes the tanning bed too much. So the environment is also going to influence our genetic expression. We can't forget that. That's important. Why, if I have the gene for breast cancer, for example, it's called BRCA1 or BRCA2, why does one person get breast cancer and one person not? Environmental expression, exactly. Not all your genetic material expresses itself. Some can be turned on, some can be turned off. So the chromosome theory of inheritance states that genes are located on specific positions or loci on a chromosome. Behavior of the chromosome during meiosis and fertilization accounts for those inherited patterns. Chromosomes that undergo segregation and independent assortment during meiosis account for Mendel's laws. So remember that whole crossing over thing? Okay. Usually the guys that kind of hang in the center tend to go together. All the guys that hang on the outside tend to what? They cross over sort independently. We're learning that. So Mendel had it down pretty good. But remember, we can have shuffling in different places and for different reasons. And remember, meiosis is going to cause that shuffling to happen, independent assortment, and give us all kinds of combinations in our gametes. Okay? So we need to understand the difference between meiosis and mitosis, and understand that the process of meiosis and the process of crossing over is going to shuffle our gene pool even more. Some genes are said to be linked. And again, they tend to be close to the center when the separation takes place. Located close together on a chromosome and tend to be inherited together. A fellow by the name of Thomas Hunt Morgan picked on many fruit flies, Drosophila melanogaster, and determined that some genes were linked based on inheritance patterns and traits. Why do you think he picked on the fruit fly? Yeah, they multiply so fast, so we can look at the offspring. And they make so many offspring. So it's not like humans. You have one, you have two, you have three. It's tough to see. Plus, they only have 2 chromosomes. Even better. So they could see all this stuff at a much faster rate.
Well, the fruit fly only has 2 girls.
I think they only have 2, yeah. That doesn't mean they only have 2 alleles. It means they have 2 chromosomes with all their alleles on there. And again, I'm not positive about that, so don't quote me on that. Anybody have a thing? thingy. They can Google that up.
Wait, what?
But they have a small number of chromosomes, so it's easier to judge.
The fruit fly on that table?
Was it on there? What did it say? I don't remember. Was it 12? It was a small number, I remember. So we can see a whole bunch of different phenotypes in the offsprings of these fruit flies at a very fast rate. And that we can determine what some of these, if some of these
It's
traits
funny
are
that
linked.
the white color is dominant because I keep dark frogs. And I have
The white color,
Yeah, is dominant.
the gray body.
Gray body.
Yeah.
same as-
Gotcha.
I keep dart frogs and I have to breed fruit flies. They're obviously flightless, but
Yeah.
yeah,
And that's
you never-
these guys.
You rarely see like the white ones.
Because look,
Yeah.
they tend to be the short-winged guys.
Well, you
And those are the flightless ones
can
that
see,
you
like,
have.
mine don't even have wings. They're,
Wingless.
yeah.
Yep.
And I mean I do have some like the white ones around sometimes, but like most of them are.
Yeah. It's not on there? We got to Google that. But also the red eyes are the wild type too. So there's tons of different mutations that geneticists can look at.
I was reading about it, and like I said, if you ever have fruit flies in your house, to watch out if one gets in your cultures, it'll just explode and all of them will just be flying, and it would be terrible.
Mm-hmm. It is a pain. And they, anybody
You never get rid of it.
Anybody who has bananas at their house in the summertime know. It's like, oh, where did they all come from? So crossing over can separate linked alleles, produce gametes with recombinant gametes, and produce offspring with recombinant phenotypes. Shuffle up the gene pool. Crossing over. Understand that concept. And again, tends to happen at the ends of homologous chromosomes. What's a homologous chromosome? Has the same what on them? Alleles. So these two guys are different chromosomes, but they have the same alleles on them. Homo means what? The same. So early studies of crossing over performed on the fruit fly, Mr. Sturtevant and Mr. Morgan. They did some more work, and they came up with these maps called linkage maps. Through lots and lots of different breedings and looking at the different genetic traits from these breedings, they could see that linked chromosomes tend to do what? Hang out in the center. And the ones that are independently crossed tend to hang out on the ends. Sex chromosomes don't just influence sex. There's other traits on those too, because they're huge. So humans have a pair of sex chromosomes, X and Y. If you're a male, you have one X and one Y. If you're a female, you have XX. And look at the letter X and look at the letter Y. There's something missing on the letter Y, yes? So we see X and we see Y. Yes? I drew it that way on purpose. Yeah. So there's a whole bunch of information missing on the Y chromosome. In females, you have 2 sets of that information. In males, you only have 1 set of that information.
Now, am I wrong by saying that the mitochondria, you said mitochondria is only on female?
The genetics of mitochondria, because mitochondria have their own genes, even in your males, come from your mother. So mitochondrial DNA is only handed down by the mother. When you look at...
Would that be that extra little,
No. Totally different.
okay.
So when you look at the reproductive cells, and I'm kind of exaggerating it, but hopefully it hits the point home. This is the egg, and this is the sperm. It's true.
What are you trying to say?
No, you saw it in the beginning. I think it was the beginning of this chapter.
Yeah, but she said, please don't put us behind the curtains.
Oh. Should have thought to do that. So who do you think carries all the organelles?
Egg.
Yeah, this one. So here's the mitochondria, here's the endoplasmic reticulum, here's all the other stuff in this big, huge cell, and this is just basically carrying what? Yeah, the DNA and a couple of little things. Once it gets in, the DNA will combine, and that big cell will then be your new you.
I know.
Yeah.
That's now a zygote.
That's now a zygote, yes, after fertilization. And after it multiplies for a few days, we start to call it then an embryo. And then the embryo for the first couple of months, after that, we call it a fetus.
And then after that,
Sounds to me like the thing is alive before it was even alive,
oh God, don't leave us
And we're not going there. Yes? So,
It wasn't a dead sperm.
that's right. But it was kind of half a cell. So it couldn't survive on its own as an individual. It's only going to survive for a short period of time. This is when we start talking about surviving for long periods of time, when the two come together. So males, females, 44. Yep. How many pairs? 22 pairs of autosomes and one pair of sex chromosomes, 2 of them. So who determines the sex of the child? Yep.
Dad.
Henry VIII, son of a gun. What did Henry do that was such a pain for all of his wives?
Cut their head off.
Because they didn't what? They didn't give him a son. Well, Henry was the problem, not his wives. Right?
Yeah.
What are the chances of producing a male?
1.4.
50-50. Yes? So whose problem was it?
His.
His problem. And he cut their heads off. So the male is the one that determines the sex of the offspring. Okay?
How many wives did he kill? 6.
I don't know. It was more than one. Too bad he didn't talk to Mendel. Or at least his wives should have talked to Mendel. Hey, it's not my fault. So you can see it's his fault. Then Mendel would have gotten his head cut off. So see the X chromosome? So much more information than the Y chromosome. So there's some traits on there. We call them sex-linked traits. Those cute little children. Whoa. That was a repeat. Some traits are located on the X chromosome. Things like color blindness. You have several different types of cones in your retina, and that's what allows you to see the range of colors that you do. Sometimes we have a genetic deficiency in producing some of these cones. Again, on the X chromosome, we're going to find this information. Who tends to be colorblind more? How come?
It's like from the Y, right? Oh, the X.
It's on the X chromosome. And women have what? 2 chances to inherit a normal gene to make a normal cone. But men only have what? 1 chance. Yes? Makes sense? Guess what else is on the sex link? What else is a sex link trait? What number is that in the middle?
7.
Can anybody not see that number?
I can see it, it's kind of difficult to see.
Yeah, it's not really bright. It's easier if you look on your book. Look at your book. Can you see, everybody see it?
Oh, yeah.
See,
Gotcha now.
yeah, it kind of gets washed out with the light. But someone who is colorblind wouldn't see that number 7. A lot of people don't realize they're colorblind because when you go to kindergarten and you learn the colors, yes, this is blue and this is red. Does that mean everybody sees the same blue and red? You might see a different shade that says, oh yeah, that's blue, even though you're not even seeing blue. Or you might see a different shade that you identify as red, but you might not be seeing red. Does that make sense?
Wait, is anybody colorblind in here?
Yeah. It's always nice when I have a colorblind one, but no. So, other things. And again, this is Punnett squares with different crosses. We have an unaffected individual, someone who's a carrier. And who would be a carrier in this case? It would be a female, right? And somebody who's colorblind. They'd have to be in 2 cases. Can females be colorblind? Sure, but they have to be what?
Carrier,
They have to have what?
2 recessive.
2 recessive alleles in order to be colorblind. So, a carrier female, and the colorblind is little n, yes? And a colorblind male can have a colorblind female and a colorblind male. Yes? So females can be colorblind, but if you look at these different scenarios, how many of these individuals who are colorblind are females? Only 1. There's more males, right? So because males only get dealt 1 copy, females get dealt 2 copies. So there's a higher incidence of colorblindness in males than there is in females. Because even if I have 1 copy, I can still produce the proper pigments and proper cones.
So how many males on that page are colorblind?
2.
Just
Versus 1.
I don't see.
So it's twice as, yeah, because of the match. See, that male, no matter what, is going to get dealt a normal gene on the X chromosome. So no colorblind males in this case. The only time we're going to see it is when there's a possibility of getting a colorblind end.
Well, how come? Oh, it's a small n?
Yeah.
Okay, because there's a...
So that's a colorblind X little n.
Okay.
So we see in all the scenarios we can have, we see twice as many males can be colorblind as females.
Colorblind is a recessive trait.
Correct. It's a sex-linked trait. So for females who get dealt 2 copies, it's a recessive occurrence. But males, you only get 1 copy. That's why they don't call it dominant or recessive. They call it sex-linked. Huh? It is or it ain't with males. Correct. Okay? Hemophilia is another sex-linked gene. And we saw the prevalence of it in a family in Europe that tended to breed a little closely. And we saw that picked up. A Russian group of people. Why can't I think of their names? Anyways, we saw it in their families. And oh, yeah, go ahead. Does it say it?
Alexandria.
There it is.
Tsar Nicholas of Russia.
That's the group. So they tended to breed a little closely. So we saw this sex-linked trait come out in their families. So it's a sex-linked recessive blood clotting trait. results in difficulty making blood clots due to genetic disorder. And it plagued the family because that mistake in their genetic code was enhanced. Because what didn't they do? Yeah, they didn't mix up their gene pool quite nicely, so it tended to plague the whole family. Dogs. Different canines got separated, eventually became inbred, and different traits were selected for. Studies indicated that small dogs were developed with the first permanent agricultural settlements in the Middle East 12,000 years ago. So we started selecting out traits that many years ago. We still do that today. Anybody breed dogs? We're not changing their genetics. We're just selecting for certain traits. What are their ancestors? From the wolf. But I look at a wolf and I look at this guy or that guy, and I see a whole bunch of different things. A Chihuahua, that one should be on the list. These are all bigger dogs. They're so different than, yeah, yeah, little tiny, short little legs, big long ears. Where the heck did that come from? That was a mistake, but somebody thought it was cute. Or things like bulldogs, for example. The way their faces are. Why do you think they're like that?
They're cow herders.
And what do?
They use their bottom jaw to break.
Exactly. Or they grab onto things and they can still breathe because their nose is back here. Yes? So that trait was desirable for whatever they happen to be using them for at the time. So when that all of a sudden popped up and the littler dogs are like, hell, that's going to work great. Take that puppy and make more. Yes? And that's where we get all the different breeds of dog. They're all canine. They're all still from the same group. Well, it's a cute puppy. Okay? So all of these cute little pictures, all these Punnett squares, all of these wonderful little terms are something we should know. Look at those cute ears on the kitty. Yeah, that particular cat has a genetics that cause its ears to do that. I don't know what it's called, but...
Is there a certain type of cat that like all have ears like that, or is it just that cat?
It's a certain breed, I guess. Curl cats? Is that what it's called?
Yeah, it's developed a curl cat mutation.
So that's the end of chapter 9. Yes? A lot of stuff in chapter 9.
It's gonna be a long quiz.
No, it's still going to be 10 questions, multiple choice. I don't know, what do you want to do? No, you have to have a test. Here's my proposal to you. Since you decided to show up to class and the rest of your classmates didn't, you get to vote. Do you want to test on 3 chapters?
Oh yeah.
Okay, or do you want to include chapter 10?
No.
Chapter 10's lots of stuff.
Yeah.
Okay? So, your next test is going to be on chapters what?
7, 8, 9.
7, 8, and 9. Now you get to choose. When do you want it?
No, that's not.
Not going to give you that option. So here's the options, and I'll give you 2 options because again, it's a lot of material, and I know, but I don't want to give you too long. So it's either going to be Monday or Wednesday.
I take Monday on because we're gonna start this chapter.
Yeah.
Yep. And we're going to start it anyway.
Yeah, but,
If we do it Wednesday, then we're gonna be listening to all of chapter 10 before we take the
No,
test.
for sure Monday. I say
Okay, vote. Who wants to take the exam on Monday? 1, 2, 3, 4, 5, 6. You're all voted. Yep. So Monday, exam on chapters. What?
7, 8, 9.
7, 8, 9. Okay.
So still a quiz on.
Yep. I'll still put it up. Photosynthesis. That was shorty. That was a shorty chapter. That's why sometimes I do 7, 8, 9, and 10, because photosynthesis is kind of a shorty chapter. And you already kind of got tested on it the last test. Okay, so the vote is Monday.
Do we get to leave now?
Nope.
I was gonna do a half an hour to 10.
Well, you know what? Okay. I might throw that to you.
You shouldn't confuse it with chapter 10 right now.
No, we're gonna have to do chapter 10 on Monday. Oh, no.
Not today, I mean.
All right, yeah, go away.